You probably knew that inside every living being, inside every plant and bacterium, there is DNA. It's a special molecule that stores biological information. But did you know that you can extract it and see it with the naked eye at home? We decided to show you how to do it and also to taste the DNA. It's easiest to extract DNA from strawberries or bananas. They are soft and have quite large cells. And DNA is of course located inside the nuclei of the cells. The first thing we needed to do was break down the cell walls of our subjects. And it's easier to do than it seems, because plants, unlike animals, have cells that are just huge. Let's say that in citrus fruits they are so large that you can separate them from each other with your hands and eat them separately. Write in the comments if you've tried it. I'm not the only one who did this, right? We mashed the strawberry and banana into a smooth puree with a fork. By the way, about the banana, to make sure the banana puree is of good quality, we invited our main banana expert, Banana Lilu. Even though the cell walls are now broken down, the DNA is still hidden in their nuclei, and the nuclei are still surrounded by cell membranes. Both of them consist of fat, and to dissolve them, we'll use regular dish soap. Distilled water is the best choice for the base of the soap solution. But first we will add salt and baking soda to our solution. This is necessary so that when the DNA is released from the cell nuclei, all its molecules stick together right away. In their normal state, protein molecules are spaced apart from each other, but when you add salt, their charges change, and they start to attract each other. Next in the recipe is a children's dish soap. The children's soap was the best choice because it was colorless and odorless. We mixed the entire solution thoroughly, and now it's time to extract the DNA from the cells. Although, actually not yet. First, this solution needs to be cooled very well. And so, it went into the freezer for half an hour. We slightly forgot about our solution and it started to turn into ice. But after a couple of minutes under the lamp, it melted back. We started with strawberry puree and added about a spoonful of this mash to 15 milliliters of the solution. And now we've reached the most important stage of the experiment. To make it all work, you need to mix this mixture very vigorously for several minutes, until as much DNA as possible is released and sticks together. The next step is to filter the resulting mixture. We used a chemical filter for this purpose, and by the way, we regretted it. Because man, that took forever. While the strawberry solution was slowly dripping through the filter, we managed to prepare the same thing with a banana and filter it. Actually, it was done through a disposable kitchen towel. It was much faster that way. By the way, notice how cool it is. The falling drops of the solution initially stay on the surface and only later merge with the main volume. After removing the unnecessary soapy foam, we moved on to the final stage of the experiment. This solution already contains the merged proteins with the DNA molecules. But in order to separate them from the water and fruit juice, we need pure ice-cold alcohol. Very carefully, using a pipette, we poured the alcohol onto the surface of the solution. Alcohol is a bit lighter than water, so it sits on its surface, just like those drops. DNA molecules do not dissolve in alcohol, so when it is added, they immediately precipitate and become visible. Then we carefully twisted the tip of a pencil at the boundary between the alcohol and the solution. And after a few seconds, a slippery clump started to form there. By hooking this substance with a needle, we managed to get it out. It's nothing more than DNA with some proteins. Indeed, judging by the color, we pulled out a bit of strawberry solution along with the DNA. The same thing happened with the banana solution. The extracted substance was yellow in color and could not be tasted in this form. At first, we decided to rinse it in distilled water, but if you were listening closely, I mentioned that DNA dissolves in water. Therefore, we extracted the DNA from the banana again, and this time, placing it on a chemical filter, we rinsed it with alcohol. In the end, it turned out to be something resembling cottage cheese. So we extracted the DNA from the banana again, and this time, placing it on a chemical filter, we rinsed it with alcohol. In the end, it turned out to be something resembling cottage cheese. We molded it into a single piece and left it to dry. After a few hours, all the alcohol and water had evaporated, and we tried a bit of the DNA, And it's a pretty strange thing. It instantly dissolves on the tongue and leaves an unpleasant, sour salty taste. In general, DNA is an acid, so a sour taste makes perfect sense. And the saltiness was probably because some salt ions combined with the proteins, the surrounding DNA molecules. Thank you so much if you watched up to this point. The video turned out to be not very long, but it's already just around the corner. Indeed, a big video about a huge terrarium. There's just a ton of footage there. Give a thumbs up and make sure you're subscribed to the channel, and then there will be even more videos like this.